Hi everyone, my name is Samantha Giannantonio, and today I'll be presenting my honors thesis titled Testing for Smad Requirement in Wnt Signaling in Drosophila or fruit fly embryos. This presentation is going to focus on two signaling pathways, but before I talk about those two pathways, it's important to establish what a signaling pathway actually is. Now, cells are going to communicate via signals, and these signals are going to activate a signaling pathway. The signal is a ligand, which is going to bind to a receptor. This binding is going to activate components in the cell's cytoplasm to enter the nucleus, bind to a response element, and that binding is then going to initiate or activate some sort of genetic response. The first signaling pathway that I'm going to talk about is Wnt signaling, which in Drosophila establishes areas of naked cuticle. Now what this means is if you look at the wild-type exoskeleton of a Drosophila embryo, between these gray shaded regions is what is called naked cuticle. Now these gray shaded regions are known as denticle belts, which are tooth-like structures that are on the belly portion of the larvae that help the larvae crawl around its environment. Now when the Wnt signaling isn't activated, then what we see is along the ventral portion only areas of denticle belts and no naked cuticle established. So Wnt signaling begins with binding of the ligand wingless to the receptor, activating the cytoplasmic components to enter the nucleus, and the response element, which is beta-catenin and TCF, are going to activate the response of forming naked cuticle. The second signaling pathway that I'm going to talk about is BMP signaling pathway, which in Drosophila establishes the dorsalmost portion of the embryo. Now, in the wild-type embryos, we see the denticle belts only along the belly portion of the embryo. While when BMP isn't active, there's no signal that tells those denticle belts to only stay in the belly, and they end up wrapping around the whole embryo, reaching into that dorsal portion. So BMP begins with the ligand DPP, binding to a receptor, activating the cytoplasmic components, which is the protein MAD, to enter the nucleus and bind to the BMP response element. So the MAD complex is going to activate that genetic response of only keeping the denticle belts in the ventral portion of the embryo. For the past 20 to 30 years, the BMP and Wnt signaling pathways have both been researched and thought to be separate and independent signaling pathways. Well, a new model recently proposed by Dr. Edward Evers in his laboratory proposed that a BMP component, MAD, acts in the Wnt signaling pathway. Now, he proposes that when MAD is not phosphorylated, meaning that a phosphate molecule isn't attached to the MAD protein, MAD actually functions as part of the Wnt response element with beta-catenin and TCF. But when MAD is phosphorylated, then it's going to continue in the BMP pathway as normal. The hypothesis that my experiment is going to test is testing the prediction that Dr. Evers made that eliminating MAD from the embryo will produce both a wingless null phenotype and a BMP null phenotype. So what this means is in the DPP null, we see denticle belts wrapping around the embryo, and in wingless null, we see no areas of naked cuticle between the denticle belts. So if he's correct, then what we would see in a MAD null phenotype is an area of denticle belt that's reaching both ventral and dorsal and no area of naked cuticle between whatsoever. So just an embryo that's completely covered in denticle belts. So the results that Evers obtained to support his hypothesis is that when he eliminated MAD from the embryo, he noticed areas of denticle belt fusion, or areas without naked cuticle between the denticle belts, which is shown in this bright yellow box. Now, this same phenotype is exhibited in a wingless loss of function phenotype when the Wnt signaling pathway isn't activated, where we see no areas of naked cuticle in panel D. Now, what this meant is that MAD could potentially function in the Wnt signaling pathway because we're seeing the same phenotypes when MAD isn't present and when wingless isn't present. One of the critiques of Dr. Evers' experiments is the way in which he eliminated MAD from the embryo. And in order to understand this first, we must understand the MAD wild-type allele and what that looks like. So the wild-type allele of MAD has a DNA binding domain, and a protein-protein interaction domain, and these two domains are joined together by a linker protein. Now, what Evers used to eliminate MAD is what is known as an RNA interference, and this is going to cut a portion of the MAD gene to eliminate the MAD protein from functioning in a signaling pathway. There are a few problems with RNA interference in that it's not entirely successful, and there still could be functional protein that could end up in a signaling pathway. Well, my experiment uses a MAD knockout allele, which eliminates the MAD gene from the embryo using genomic deletion. So in order to make a MAD protein for a signaling pathway, it begins with the MAD gene, which we saw in the previous slide, and the MAD gene is going to be transcribed into an intermediate, which is a messenger RNA. And this messenger RNA is going to be translated into a functional protein made for a signaling pathway. Now, when RNA interference plays a role, the MAD gene is still going to exist, but RNA interference is going to eliminate that middleman, which is the intermediate of the messenger RNA. And using RNA interference is only 50 to 90% effective. So hopefully, no functional protein was made for a signaling pathway, but it's really difficult to know because of the low success rate of RNA interference. Now, a more definitive method to eliminate MAD from the embryo is what is known as a gene knockout. And this method wasn't available for Dr. Evers, but it's available now thank you to Dr. Sheila Mosali in our lab, who created a MAD knockout allele. Now, what this does is eliminate the MAD gene in its entirety. So no mRNA can be transcribed, and therefore no protein can be translated because there's not even a gene to work with in the first place. One of the challenging components about MAD is that it's not a protein that's only present in the embryo, but it's also preloaded into the oocyte or the egg. So MAD mRNA is going to be loaded into the oocyte by mom, and upon fertilization, the MAD mRNA is translated into protein. So now the developing embryo is going to have protein from mom while also making its own mRNAs for MAD. So when you're looking at an embryo, a mature embryo, there's going to be components from mom as well as components the embryo made itself. And so the strains that I needed to create for my experiment are homozygous MAD knockout, meaning that MAD was completely eliminated from the oocyte as well as the embryo to ensure that there's no MAD protein involved in the embryo whatsoever. So in order to better understand the phenotypes exhibited by my experimental homozygous MAD knockout strain, I have reference embryos for comparison. And so the embryos that I have for reference are a wingless null embryo, a DPP null embryo, and just a wild-type embryo to see what denticle belts should normally look like. And these embryos are going to be scored on a scale of 1 to 3, 1 being the most wild-type looking, and 3 being the most like that phenotype of the reference embryo. So the scoring scale and the reference embryos are really useful when looking at predicted results of this experiment. So if Evers' hypothesis is correct and MAD does function in both the BMP and Wnt signaling pathways, then what we would see in a homozygous MAD knockout embryo is that it would look both like a DPP null allele and a wingless null allele, indicating that MAD does function in both signaling pathways. If Evers' hypothesis is incorrect and MAD only functions in the BMP signaling pathway as previously thought, then a homozygous MAD knockout allele will only look like a DPP null allele, indicating that MAD is only present in the BMP signaling pathway. Now, the results of this experiment are still ongoing due to some unexpected weaknesses with the homozygous MAD knockout strains. However, I'm continuing to build these strains in order to obtain the appropriate number of flies. But I do have some results that are actually throughout this presentation. If you look at my DPP null image and my wild type image, these are images that I've obtained from my reference embryo cuticle preparations. So there are still results that I am obtaining, and it's really important to obtain these images to compare my experimental strains to. I hope to be finishing this experiment in the next few weeks, but I thank you so much for listening to my presentation, and I'd like to thank those listed on this slide for their contributions in my honors thesis.